Hi viewers, this is J. Swami, Assistant Professor of Zoology. In this class, we are going to discuss the structure of skeletal muscle. Generally, skeletal muscles are composed of skeletal muscle cells, which are called as muscle fibers. These muscle fibers are always in association with the connective tissue, blood vessels, and nerves. In general, each muscle fiber, each muscle that is covered by the three connective tissue layers. Three connective tissue layers, namely epimysium, perimysium, and the endomysium. There are three layers which cover the muscle. These connective tissue layers are called as the epimysium, perimysium, and the endomysium. What is the epimysium is? Epi means on, above. On muscle. Mysium means muscle. This epimysium is a dense layer of connective tissue consisting of collagen fibers that surrounds the entire muscle. Whereas the perimysium, if you observe, peri means around. The perimysium divides the skeletal muscle into a series of compartments. They are called as a fascicle. Fasciculus is a single bundle. Fascicles, plural. Each and every fascicle, it contains of about 10 to 100 muscle fibers. 10 to 100 muscle fibers. In addition to collagen and elastic fibers, the perimysium contain blood vessels and nerves that serve the muscle fibers to the muscle fibers of the fascicle. Whereas if we come to the endomysium, endo means inside. Endomysium is the connective tissue layer that surrounds the individual muscle fibers and loosely interconnects adjacent muscle fibers. This flexible elastic connective tissue layer contains capillary networks that supply blood to the muscle fibers, nerve fibers that control the muscle, myosatellite cells, tissue stem cells that take part in the repair of damaged muscles. If you observe furthermore, here you can see this is the epimysium, perimysium, and the endomysium. So finally it gives rise to the formation of a myofibril, which is composed of thick myosin filaments and the thin actin filaments. At each end of the muscle, the collagen fibers of the epimysium, perimysium, and endomysium interweave and form either a bundle known as the tendon. It is known as the tendon, or a broad sheet-like structure which is called as aponeurosis. Both the tendons and the aponeurosis, they attach skeletal muscle to the bone. Where they contact the bone, the collagen fibers extend into the bone matrix, providing a firm attachment. As a result, the contraction of the muscle pulls on the attached bone. Basically, some of the muscles, however, they are not attached to the bones. For example, sphincters that guard the certain openings, muscles of tongue and abdomen, they may not attach to any kind of bones. This is the external structure of the skeletal muscles, where you can find out the epimysium, perimysium, and the endomysium. Coming to the ultrastructure of the skeletal muscle fiber. The skeletal muscle is composed of hundreds to thousands of cells, which are called as the muscle fibers. Otherwise the myofibrils. These muscle fibers, they are looking in the elongated shapes. The typical length of a mature skeletal muscle fiber is of about 10 centimeters. Some of them, they are as long as 30 centimeters, which are present in the muscles of thigh. The diameter of a mature skeletal muscle fiber ranges from 10 to 100 micrometers. 10 to 100 micrometers. The muscle fiber is covered with plasma membrane called the sarcolemma. The cytoplasmic membrane which is surrounding the muscle fiber is known as the sarcolemma. And the cytoplasmic fluid which is present inside the muscle fiber, otherwise the cytoplasm of the muscle fiber, that is known as the sarcoplasm. The nucleus of the muscle fiber is known as the sarcode. The mitochondria of the muscle fiber is known as the sarcosome. And the endoplasmic reticulum, which is present inside the muscle fiber, is known as the sarcoplasmic reticulum. So the cytoplasmic covering of the muscle fiber is sarcolemma. Cytoplasm of the muscle fiber is the sarcoplasm. Nucleus of the muscle fiber is the sarcode. Mitochondria of the muscle fiber is the sarcosome, right? And endoplasmic reticulum of the muscle fiber is the sarcoplasmic reticulum. So these are the different terms which are frequently used in the structure of the muscle fiber. And here you can also furthermore you can see there is a triad system. What is a triad system is thousands of tiny invaginations of the sarcolemma called as the T-tubules, otherwise the transverse tubules. T-tubules are transverse tubules that made by the invagination of the sarcolemma, that is a tunnel-like structure will be formed from the surface of the muscle into the center of the muscle. A tunnel-like structure has been formed by the invagination of the sarcolemma from the surface of the muscle fiber into the center of the muscle fiber. That is, each and every T-tubule, it open to the outside of the fiber. They are filled with the interstitial fluid. Muscle action potential travel along the sarcolemma and through the T-tubules, quickly spreading the action potential throughout the muscle fiber. This kind of arrangement ensures that an action potential excites all the parts of the muscle fiber at essentially the same instant. Then if you observe, a T-tubule is present in the middle. On either sides of the T-tubule, you can find out the terminal cisternae of the sarcoplasmic reticulum on the either sides. So a transverse tubule and the two terminal cisternae of the sarcoplasmic reticulum on both the sides, it form a triad system. It is called as a triad. T-tubule and the terminal cisternae of the cytoplasmic reticulum or sarcoplasmic reticulum. All these three structures, they form a triad. In a relaxed muscle fiber, the sarcoplasmic reticulum is filled up with stores the calcium ions. Then the sarcoplasm contain the organelles like the sarcosome, which is called as a mitochondria, which lie strategically close to the contractile machinery that uses the ATP during the contraction, so that ATP can be produced quickly as needed. The sarcoplasm also contains glycogen granules, myoglobin, found only in muscle. So just like the hemoglobin which we will see in the blood, like that there is a myoglobin that found only in the muscle that binds to the oxygen molecules that diffuse into the muscle fibers from interstitial fluid. Myoglobin releases oxygen when it is needed by the mitochondria for ATP production. And if you see furthermore, at the high magnification under the microscope, the sarcoplasm appears stuffed with the little threads. The small structures are the myofibrils. Myo means muscle, fibrilla means little fiber. The contractile elements of the skeletal muscles, the myofibrils are of about. So this, each and every one is a myofibril. So these all are the myofibrils. These myofibrils are of about 2 micrometers in diameter and extend the entire length of the muscle fiber. Their prominent striations make the entire skeletal muscle fiber appear to be striped or striated. Myofibrils that contain two kinds of myofilament, they are called as the thick filament and the thin filament. Thick filament and the thin filaments. Let us see about the thick filament. Here this is the thick filament. Thick filaments are 16 nanometers in diameter and 1 to 2 micrometers long and compose mostly of the protein which is called as the myosin protein, which functions as the motor protein in all the types of muscles including the smooth muscle. This motor protein pulls various cellular structures to achieve the movement by converting the chemical energy into ATP to the mechanical energy of motion, that is the production of force. In skeletal muscle, about 300 molecules of myosin join to form a single thick filament. A single thick filament consists of about 300 molecules of the myosin. Here you can observe furthermore the thick filament. So here this is the thick filament. Here the myosin molecules have the tail region. Myosin molecules have the tail region and head region. Tail region and the head region. Here the tails of the neighboring myosin molecules, they are arranged parallel to one another, forming the shaft of the thick filament. So these all are the tail regions of the myosin molecules. Whereas each myosin molecule, the molecular weight of the myosin molecule may be of about 4,60,000 daltons. 4,60,000 daltons. So these molecules, they have the two regions. One is the tail, another one is the head region. Tail region and the head region. Tails are twisted and parallelly arranged and forms the shaft-like structure, whereas the heads of the molecule, they look like the two golf clubs. They are twisted together, looks like, and made up of two types of myromyosins. Simply we can say there are two kinds of myromyosins, heavy myromyosins and the light myromyosins. Light myromyosin, that is the tail region. Heavy myromyosin, that is the head region, HMM or LMM. Light myromyosin and the heavy myromycin based upon their molecular weight. That is, the myosin molecule get divided into two portions, light myromycin and the heavy myromycin. Let us see one by one the light myromycin. It is the tail or backbone portion of the myosin molecule which intertwines with the tail of the other myosin molecules to form a thick filament. Whereas the heavy myromycin HMM has the terminal double head projecting laterally from one end of the each molecule and extending towards one of the six thin filaments that surround each thick filament. The head contains specific binding sites. Specific binding sites. These specific binding sites give accommodation. They give accommodation for the attachment of the myosin molecule with that of the actin molecule, and there is a ATP receptor site. Here we can observe one is the actin binding site on the myosin, and there is a enzyme which attached to this myosin ATPase site. Myosin ATPase site, and there is also a ATP receptor site. So that is, ATPase is the enzyme which hydrolyzes the ATP in order to release the energy for the contraction. So here the heads, they have the specific binding sites for the formation of the actomyosin complex with the addition of the actin, and there is a ATP receptor site and a catalytic ATPase site, and thereby the ATP get hydrolyzed and that gives the energy for the contraction. Then coming to the thin filaments. Thin filaments are of about 8 nanometers in diameter, 8 nanometers in diameter, and 1 to 2 micrometers long, and that contain a contractile protein which is called as the actin. Here the contractile protein actin, it is, if it is singular, it is a globular actin. If the globular actin undergo polymerization, that forms a fibril or actin, F actin. G actin is the globular actin, and that is, individual molecules, actin molecules are called as the globular actin. They join to form a filament like actin molecule, that is the F actin, that is twisted into a helix. And along with the protein actin, there is a small amounts of regulatory proteins like the tropomyosin and the troponin. These are also present in the thin filaments. Thin filaments are 8 nanometer in diameter and 1 to 2 micrometers long, that contain the contractile protein actin along with the two regulatory proteins like the tropomyosin and the troponin. Here the actin, actually it is in the form of a globular actin, and if it is polymerized to form the filamentous actin or F actin. On each actin molecule, here there is a myosin binding site where a myosin head can attach to that of the actin. Then if you come to the tropomyosin, here, so the green color one, tropomyosin is a long thread-like helical protein molecule and lies on actin filament that covers, masking the myosin binding sites of the actin. This thread-like structure, it covers the myosin binding site of the actin. If we come to the troponin, has 3 subunits: troponin T, troponin C, and troponin I. Troponin T binds to the tropomyosin, and troponin C binds to the calcium ions, and troponin I, whenever it is necessary, it inhibits the contraction. It is a inhibitory. Troponin I is the inhibitory subunit. In a relaxed muscle, myosin is blocked from binding to actin because strands of the tropomyosin, they mask the myosin binding sites upon the actin. The tropomyosin strands in turn, they are held in a place by troponin molecules. When the calcium ions bind to troponin, when the calcium ion binds to troponin, it undergoes a change in shape. This change moves tropomyosin away from the myosin binding sites on the actin, and cross bridges are being formed. Then the heavy myromyosin of the myosin binds to the binding sites on the actin. Binding sites on the actin. Then here you can see actin formed by three different proteins. That is globular actin, that may form into, see here it is the individual actin, it is a filamentous actin or F actin. So these black spots are the myosin binding site upon the actin. Myosin binding site upon the actin. You can see here, this is a thread-like, pink color thread-like structure is the tropomyosin, and here it is the pink color ball-like structure troponin complex, which is composed of three subunits: troponin T, troponin C, and troponin I. In general, so whenever it is in the resting condition, these myosin binding sites upon the actin, they masked by the tropomyosin thread-like protein. So whenever the calcium ions comes in contact with the troponin molecule C, troponin C, then that will move like this. So the myosin binding sites upon the actin, they will be opened, and thereby there is a attachment in between the head region of the myosin and with that of the myosin binding site of the actin. That leads to the muscle contraction. Then coming to the ultrastructure of the muscle fiber or myofibril. Myofilaments are joined end to end to form the myofibrils and form highly ordered repeating units called as the sarcomeres. They are called as the sarcomeres. The sarcomere is a structural and functional unit of the muscle, and that is responsible for the muscle contraction. If you see the arrangement of the sarcomere from the adjacent Z lines, the region in between adjacent Z lines is a single sarcomere. This portion is a single sarcomere. The arrangement of the actin and myosin filaments gives the myofibril a banded or striated appearance with dark A bands, alternating with the light I bands. Here you can observe, this is the A band, dark A band, and these are the light I bands. The A bands have a central H zone. In the middle of the A band, you can find out a central H zone, which is again divided by a faint M line. A faint M line is present in the middle of the H zone. And if you observe the I band, they are also divided by a disc-like structure which is called as a Z disc. In the middle of the I band, you can also see a disc-like structure which divides the I band. So actually it is the Z disc or Z line is a filamentous network of protein forming a disc-like structure for the attachment of the actin filaments. Now if you define the sarcomere, here you can see sarcomere is defined as the segment between two adjacent Z lines within a myofibril. So here there is a Z line and here there is a Z line. So the region in between these two adjacent Z lines is to be considered as the sarcomere. The I band is lighter, less dense area on each side of the Z disc and contains only actin thin filaments which extend into the A band among the thick myosin filaments. Here you can observe the I band. It's made up of the actin filaments. These actin filaments, they move inside to the A band. They move inside to the A band, right? Then the darker middle part of the sarcomere is the A band. The darker middle part of the sarcomere is the A band, which extend the entire length of the thick filaments. The actin and myosin filaments overlap at both ends of the A band, producing a darker appearance. Each myosin filament is surrounded by 6 actin filaments, where actin and myosin filaments overlap. In the middle of the each A band is a lighter area which is called as a H zone. This lighter area is known as H band or H zone, where only myosin filaments are present. Where only myosin filaments are present, right? Then here you can see in the I band only actin filaments. In the A band, in the H region only myosin filaments, but the remaining portion both actin and myosin filaments. The navy blue color, royal blue color is the myosin filament and red colored one is the actin filaments. That is, in the middle of each A band is a lighter area called as the H zone, where only myosin filaments are present. In the middle, a dark line called the M line is in the middle of the H zone and consists of delicate filaments that attach myosin filaments to the center of the sarcomere. The M line helps hold the myosin filaments in place, similar to the way the Z disc holds actin filament in place. So here, if you observe, the A stands for A band, means an isotropic band. Dark region, dark bands. I band means isotropic band. The bands are much less birefringent than the A bands. And H stands for heller. The meaning of heller in the German is the bright. Whereas M stands for the mittel, M-I-T-T-L, mittel. The meaning of the mittel is the middle in German. And if you observe the Z, it is a Zwischenschieb. The meaning of the Zwischenschieb is the band in between. Band in between. So anisotropic, isotropic, heller, mittel, and the Zwischenschieb. These all are the terms which are usually used while we are going for the muscle contraction. Here you can see the Z discs. They are narrow plate shaped regions of dense material that separate one sarcomere from the next sarcomere. That is, so here this is the entire one sarcomere. So this is another adjacent one, this is another sarcomere. A band, what is the specialty of the A band? Dark middle part of sarcomere that extends entire length of thick filaments and includes those parts of thin filaments that overlap thick filaments. Coming to the I band, lighter, less dense area of sarcomere that contains remainder of the thin filaments but no thick filaments. A Z disc passes through center of the each of I band. Then coming to the H zone or H band, narrow region in center of each A band that contains thick filaments but no thin filaments. M line, region in center of H zone that contains proteins that hold thick filaments together at center of the sarcomere. So these all are the vocabulary that related to the structure of the muscle fiber. So here thick myosin filaments, thin actin filaments, and here you can observe. So this is a single sarcomere. So this is one, this is another sarcomere, this is another sarcomere. So the A band contain M line, H band, zone of overlapping, the I band, Z line. Here there are other proteins which are present in the muscle fiber, namely titin, nebulin, tropomodulin, and myomycin. Besides the contractile and regulatory proteins of the muscle fiber, the muscle also contains about a dozen structural proteins which contribute to the alignment, stability, elasticity, and extensibility of myofibrils. Important structural proteins are the titin, actinin, myomycin, nebulin, and dystrophin. Titin is the third most plentiful protein in a skeletal muscle after actin and myosin. This molecule's name reflects its huge size with a molecular mass of about 3 million daltons. Titin is 50 times larger than an average sized protein. Each titin molecule span of the sarcomere from a Z disc to an M line, a distance of 1 to 1.2 micrometers in relaxed muscle. Each titin molecule connects a Z disc to the M line of this sarcomere, thereby helping stabilize the position of the thick filament. The part of the titin molecule that extends from the Z disc is very elastic because it can stretch to at least 4 times its resting length and then spring back unharmed. Titin accounts for much of the elasticity and extensibility of the myofibrils. Titin probably helps the sarcomere return to its resting length after a muscle has contracted or been stretched, may help prevent overextension of sarcomeres and maintains the central location of the A band. The dense material of the Z disc contains molecules of alpha actinin, which binds to actin molecules of the thin filament and to titin. So these are the certain proteins, another proteins which are present, and they cause the elasticity and extensibility. So they are titin, nebulin, tropomodulin, and as well as the myomycin. They are also helping in the muscle contraction. So this is all about the structure of the muscle and ultra structure of the muscle fiber. Thank you.